Hi friends, welcome to the session. I am Supriya. Today I am here to discuss with you one of the most exciting topic in your textbook, that is biotechnology, right? So in your textbook, biotechnology unit has two lessons. One is principles and processes and the other one is applications of biotechnology. Now so friends, I would like to begin with the first lesson, that is biotechnology, principles and processes. So before getting into the topic, let us have a look at these things. So can you identify this image? This is Louis Brown, the first test tube baby. And this is Humulin, the first recombinant insulin produced by the company Eli Lilly. And you must be knowing about this Dolly, the first cloned sheep. And do you know the person here, the liquor baron of India? Yes, this is the beer, and here I have given you 4 examples. All these 4 examples are nothing but the processes of biotechnology. And we'll try to understand how all these processes are coming under the topic of biotechnology. So you can see here, and my dear friends, you all must be knowing this term biotechnology. You are not new to this, right? So the term biotechnology was introduced by Carl Ehricke, and father of biotechnology was Paul Berg. And here biotechnology is known to be the buzzword of 21st century. So to know why it became the buzzword of the 21st century, we should know what is biotechnology, what are its contributions for the human welfare. Biotechnology being an offshoot of the modern biology, it has revolutionized the way in which we live today. The major impact it has on the fields of healthcare and food production. Right? Now what is this biotechnology? Why it has become the buzzword of the 21st century and how it has revolutionized the way in which we are living today? What kind of impact it has brought in the fields of agriculture and medicine? Let us get into the details. Before we try to understand the definition of biotechnology, let us first understand what is science and what is technology. So well, my dear friends, science is a systematic study of anything which is based on observations and experimentation. So man is inquisitive in nature. Out of his curiosity, we started investigating the elements of nature, the physical, chemical and the biological elements of nature. And as a result, we started observing and experimenting with things, and there is a vast body of knowledge accumulated today. That is called as science. On the other hand, technology is nothing but application of the principles and laws of science in order to design something new, right? So technology is making use of the knowledge that we gained in science to produce a product, and this product will essentially uplift, or this product can really bring a change in the living standards of humans, right? So here, to give a few examples of technologies, we have road technology, we have building technology, we have bridge making technologies, we have paint technology, everything, whatnot. So in this current world, everything is running on technology, and technology is of course the fruit of science. And it is this technology that has given to the rise of industrialization. With the dawn of industrialization, it is the physical and the chemical sciences that have dominated the world. Then what is this biotechnology? Biotechnology is also a technology in which we are making use of biological materials as raw materials. When you compare with the industrialized world that makes use of physical and chemical technologies, biotechnology is different because here we are making use of the biological techniques, tools and processes in order to make a new product, right? So In the industrial world, physical science and chemical science happen to be the most exciting fields, whereas until the 20th century, until the end of the 20th century, biological science was considered as a dull science. Why? Because right from the times of Aristotle through the medieval periods and after the Renaissance, the post-Renaissance periods, biology was studied as the science of plants and animals. Under biology, we only tried to learn the morphology, the external features of living organisms, and we tried to classify plants and animals. And that is how the biological sciences was not considered as an exciting field of science. But with the advent of microscopy, with the invention of microscopy, it gave rise to a new world of science, that is microbiology. Now later, formulation of cell theory and Darwin's evolutionary theory, Mendelian genetics, rediscovery of Mendelian genetics, they gave birth to a new field of biology, that is the genetics, study of the DNA, study of inheritance, etc. etc. Now, after genetics, after the invention of electron microscopy, we came to understand that the ultrastructures of the prokaryotic and the eukaryotic cell. Now, the actual excitement in biology came to existence only in the late 20th century with the elucidation of the structure of DNA by Watson and Crick. Now, after the structure of DNA was given, soon it was proved that DNA is the genetic material, and later the central dogma of molecular biology and the genetic code were cracked. And this led to the exciting field of molecular biology. So as you can see here, with the advent of molecular biology, there came up the recombinant DNA technology, production of recombinant proteins like insulin, and today we are living in the era of genomics and proteomics, right? So as you can see here, only with the developments taking place in the fields of cell biology, then molecular biology, and special techniques involved in cracking the genetic code, etc., it gave rise to an exciting science of biological sciences, and that is your biotechnology. Biotechnology is essentially an applied science. As you can see here, biotechnology is drawing heavily from so many other biological branches like biochemistry, cell biology, genetics, chemical engineering, genetic engineering, molecular biology and microbiology. All these are the foundations on which the field of biotechnology is built. So, in today's class, we will be learning about the definition of biotechnology, examples of biotechnological processes, the two core areas of biotechnology, how recombinant DNA technology is superior when compared to traditional breeding techniques, right? So let us start the exciting journey into the world of biotechnology. So as I told you, biotechnology is also a technology just like any other technology. So technology is concerned with designing a product and producing it on a large scale, which will have its positive impact in improving the standard of human beings. So here, biotechnology can be understood as a collection of technologies in which the living organisms or their enzymes are used as raw materials in order to produce products and services, and these products should be produced on a large scale, which will be useful for the human beings. Yes, so biotechnology is a collection of techniques in which we are making use of living organisms or their enzymes as raw materials in order to design or produce a product which will be useful for the human beings, and this product should essentially be produced on a large scale, right? So now, according to this definition, conversion of milk into curd conversion of milk into curd, which involves the microbes, that is lactobacillus, right? So can we call making curd as a biotechnological process because it involves milk as raw material and it involves the microbial process, that is fermentation. Can we call making bread as a biotechnological process because it involves yeast fermenting the dough? Can we call wine as a biotechnological process because it involves fermenting the grape juice? Well, my dear friends, making of curd, bread and wine, these three processes essentially involve microbes, but they cannot be called as biotechnological processes. Why? Because any biotechnological process, the development of the product should be involved with recombinant DNA technologies. That is, when you want to call some product as a biotechnological product, it should be involving, the product development should involve the modification of the genes, application of the recombinant DNA techniques, etc. So for this reason, making curd, bread and wine, though they involve microbe-mediated processes, they cannot be called as biotechnologies. And in other words, in these cases, we are making use of the natural abilities of microbes. We are not modifying the microbes or anything, and for this reason, we don't call them as biotechnological processes. So here, biotechnological process refers to use of genetically modified organisms to produce the product, and you have to produce the product on a large scale. This is also essential. One is to produce the product, to design the product, you have to make use of recombinant DNA techniques, and the product should be made on a large scale. Only then you can call it as a biotechnological process. Now, let us see the definition given by European Federation of Biotechnology. According to the EFB, the definition goes like this. Biotechnology is an integration of natural science and organisms, cells, parts thereof and molecular analogues in order to produce products and services. So, read the definition carefully. Biotechnology is an integration, that means it's an amalgamation of natural science and the organisms, cells, parts thereof. Parts thereof means they are parts. And the molecular analogues. What do you mean by molecular analogues? Similar to the natural biomolecules are called as molecular analogues. So here, why did we integrate all these things? In order to produce goods and services, right? So this is the definition given by European Federation of Biotechnology. Let us try to understand the definition in a little more detail. Suppose if we are making use of the organism as a whole in order to produce a product. Let us take this example. We have a plant, and in this plant we have introduced a gene that is extracted from leech. So leech belongs to animal kingdom, and from this leech we have derived a gene that is coding for hirudin protein. And now this plant which is carrying the gene from the animal kingdom is called as transgenic plant. Transgenic plant is a plant that carries a gene from some other organism. Now from this transgenic plant is used as a factory to make the hirudin. Hirudin is the product. What is the significance of hirudin? Hirudin is used as an anticoagulant. It is having its own importance in the field of medicine. So here we have picked a gene from one organism belonging to a different kingdom, and we have placed this in another organism of plant kingdom. Now we are making use of this transgenic organism. The plant here is called as transgenic plant as a factory of making the animal product, that is hirudin, and we are extracting this product and we are selling it in the market. And in this case, Hirudin is the gene that is coding for the protein hirudin, and it is important because it is an anticoagulant and it is important product in the field of healthcare. Now, if you go for an example where we are making use of organism at a cellular level, one simple example can be the recombinant yeast. So you all know, brewing industry, beer is made from barley. Barley seeds are fermented. So from the barley we get the beer. And the organism responsible here is the yeast. Now, beer industry is one large industry, and in the beer industry they make use of the recombinant yeast. In spite of the natural yeast, they make use of the recombinant yeast, right? Now, in this process, what is the microbial process involved here? It is fermentation, right? Barley is converted into beer by the action of yeast in a process called fermentation, and here the brewing industry is using the yeast as an organism, and the yeast is essentially a recombinant or genetically modified organism. This can be an example when we make use of an organism at a cellular level. Now, if we look at usage of parts of an organism, you can take a simple example of isolated enzymes. So here we are not making use of the organism as a whole, we are not making use of a cell, we are making use of an isolated enzyme. You pick the enzyme from the cell, purify it, and make use of this enzyme to convert the substrate into a product. So this can also happen. So examples of such enzymes that are widely used in industries are proteases, pectinases in the food processing industries. Now, certain examples are given in a textbook. Let us have a look at them. In vitro fertilization and production of test tube baby comes under biotechnological process. Why test tube baby is coming under biotechnological process is because in order to go for IVF, you must have studied about IVF, right? In the process of IVF, we are actually collecting the gametes from the male and the female organisms, and we are allowing them to fuse artificially in a laboratory, right? So fertilization is supposed to happen naturally, internally, but here we are collecting the gametes of both male and female parents, and we are getting the fertilization done in a Petri plate. And what is the product of fertilization? It is the embryo. And this embryo is again transferred back into the mother's womb. In this case, because we are handling the cells, we are culturing the cells artificially in a laboratory, and we are producing a product, test tube baby is otherwise called as IVF. In fact, test tube baby is a term used by a common man. Fertilization actually takes place in a Petri plate, not in a test tube, and we have to call it as IVF, in vitro fertilization technique. Now the second case, synthesis of a gene. For the first time, yeast tRNA coding gene was synthesized in a laboratory, right? So we are able to synthesize, make a copy of a gene in a laboratory itself. Now the third one is developing a DNA vaccine. So today, vaccination is quite safe, especially the DNA vaccines are known to be the safest vaccines. So vaccine is required for immunization program on a large scale. From where do you think we are able to produce the DNA wala vaccine on a large scale? With the help of biotechnological process. Now, correcting a defective gene. So my dear friends, if a person is suffering from a disorder and the disorder is due to presence of a defective gene, just like how we can replace a defective part in a machine, the same way there is a possibility to replace a defective gene with a functional gene copy, and that is how the disorder, the person suffering with disorder can become normal. This is a possibility. This process is called as gene therapy. That is, injecting a correct gene in place of a defective gene and rectify the defect of a given person is called as gene therapy. It is a possibility now, and all these are the different examples of biotechnological processes. Now let us see what are the two core areas of biotechnology. As I told you, biotechnology is an industrial process essentially. It is the production of a product or a goods on a large scale. And how is it different from other technologies? It is different from other technologies because here we are making use of recombinant DNA technology and designing the product. Designing is essentially an engineering, right? What do you mean by engineering? Engineering is nothing but to design something new by applying the principles of science. So here, because biotechnology essentially is concerned with designing a product and producing the product on a large scale, the two core areas of biotechnology involve genetic engineering and maintenance of sterile culture under chemical engineering process. Let us see what are these two core areas in a bit of detail. So the first area is concerned with designing of the product. Genetic engineering is all concerned with design product, right? So as you can see here, in the process of genetic engineering, we are altering the chemistry of DNA or RNA. We alter the chemistry. That means we know nucleic acid molecule is DNA or RNA. Nucleic acids are polymers of nucleotides. So what we do is you take a naturally existing DNA and chop off some parts of the DNA, replace them with new bits of DNA, and that is how you are going to design a recombinant DNA or a designer DNA, right? So that means you are going to design a new molecule of DNA which was not existing in nature previously, right? So in the process of genetic engineering, we alter the chemistry of the genetic material and we create the recombinant DNA molecule. Now once we create the recombinant DNA molecule, we are introducing this molecule into the host organism. The organism which takes up the recombinant molecule is called as a host organism. Now, because of entry of recombinant molecule, the host phenotype will be changed, right? So how do we understand this? We are taking a naturally occurring DNA molecule and we are doing some modifications to it, like removing certain genes and adding new genes. Once the modification is done, the modified DNA called as the recombinant DNA is inserted into the host cell. A host is an organism into which we introduce the recombinant DNA. Once the host has taken up the recombinant DNA, you are calling this host as a recombinant host. Now this recombinant host is something different from its previous phenotype. Previously this host was not having that particular gene, and now after entry of the recombinant DNA, it is carrying a new gene, and as a result its phenotype is also changed, right? So this is all involved with designing of the product. Now in the next step, so once we got the product, the prototype of the product, that is the recombinant host, what is the next step? We have to provide the optimum conditions for the growth of the recombinant host. It is just like how a farmer provides an ideal conditions of fertilizer and water for his crop to grow in its optimum state. The same way, here we are growing the recombinant host. How a farmer is growing a crop? For what purpose he grows a crop? He wants to harvest the product. The same way here also, we are looking for some product from this recombinant host. And for this reason, we have to provide optimum conditions. Large scale growth or large scale culture of the recombinant host, large scale culture of the recombinant host. For that reason, we have to provide optimum conditions. We have to provide optimum conditions, right? So this area is essentially involved with chemical engineering processes, designing of a vessel. So in order to cultivate the recombinant host on a large scale, we need to design a big vessel, a large vessel called as a fermenter, and we have to provide nutrients at their optimum, and we have to maintain sterile conditions. What do you mean by sterile conditions? Sterile conditions means, so as you can see, free of the microbial environment. Always remember, here we are dealing with the growth of one variety of microbe only. We don't want other varieties of microbes to grow. So what we can do is, see, it is like in nature, all the organisms will exist in mixed populations. We don't find, crop is not natural, right? So in a cropland, suppose if we are growing rice, if there is any weed growing, unwanted plant growing, what the farmer does, he will eliminate the weeds, right? Here also, in nature, microbes live in mixed cultures. So many varieties of microbes will live together. But we don't want that here. We have designed the recombinant host, we have designed a special vessel, and in this special vessel, we have to maintain sterile conditions. Under sterile conditions, except for the desired organism, only the host should grow, only the host cell should grow. No other unwanted microbe should be growing in this fermenter. The special vessel in which you are growing is called as a fermenter. In this fermenter, only our desired organism should grow, no undesired organism should grow. That is called as maintenance of sterile conditions. So these are the two core areas of biotechnology. So here in these processes, we are able to produce or manufacture antibiotics, vaccines and enzymes on a large scale, right? So my dear friends, so till now we tried to understand the definition of biotechnology, what are the processes that can be called as biotechnology, right? Now, and what are the two core areas of biotechnology? Now let us try to understand how this recombinant DNA technology is much more superior when compared to that of the traditional breeding, sexual reproduction and asexual reproduction. If you can see here, when we compare the asexual and sexual reproduction, it is very clear. We all already knew these things. So asexual reproduction involves only mitotic divisions and it is uniparental inheritance, because of which the genetic material will flow from parent to offspring as it is. The progeny developed from asexual reproduction, the progeny is identical to the parent. Identical to parent. Identical means it is 100% a Xerox copy of its parent. No variations can be seen in the asexual reproduction. And in such cases, the function of asexual reproduction is to preserve the genetic information as it is from one generation to other generation. But if we see the sexual reproduction essentially involves fusion of gametes and it is biparental inheritance. And for gamete formation, which cell division is involved? It is meiosis, right? So here, sexual reproduction is biparental, gamete formation is involving with meiotic cell division, meiosis allows formation of new combinations. As a result, sexual reproduction provides opportunities for variations. And that is the reason the progeny that comes from sexual reproduction are only similar to the parents, but not identical. They are similar to the parent but not identical. We all resemble our parents, right? But none of us are identical to the parent, right? That is because we are all products of sexual reproduction. Now, when you compare to asexual reproduction, sexual reproduction is giving a chance for variations, and these variations will give rise to unique combinations of genetic setup, and these new combinations means nature is providing an opportunity to reshuffle the genes that are already present in the parents, and these reshuffling will help in formulation of new combinations. With the existing genes, it is trying to change the combinations, and these change of combinations will lead to unique combinations of genes, and this can be beneficial to the organism as well as the population, right? So, sexual reproduction provides opportunities for variations, and as a result, the variations may lead to, they can offer beneficial to the organism, and sometimes they can also be beneficial to the population as a whole. But in sexual reproduction, they cannot cross the species barrier. That means a jasmine plant can undergo sexual reproduction only with a jasmine plant. But if we are expecting a hibiscus flower with the fragrance of jasmine, it is not possible. Why? Because sexual reproduction is species specific. That means it cannot cross the species barrier. And to overcome that drawback, we have formulated a hybridization technique. So, in plant and animal breeding fields, hybridization is the technique that is employed. And in this hybridization technique, we are taking parents of related species. Two parents of related species are taken, and these two parents with selected characters are meant for sexual hybridization. So, in traditional hybridization, we are choosing the parents with desired characters and these two parents are used to produce the hybrids. Right? Now, in this case, the reproduction is sexual reproduction as usual, but the thing is that both the parents are having the desired characters. Now, the resultant progeny, the hybrid progeny, is expected to be superior when compared to its parent, right? So, this is how we are able to formulate the traditional hybridization in the fields of plant and animal breeding. And as a result, we are able to develop new varieties. But traditional hybridization has its own drawbacks. What are the drawbacks of traditional hybridization? During hybridization, both undesirable and desirable genes can enter into the progeny. So, as you can see here, though we have chosen the parents with desired characters, along with the desired characters, the progeny cells can also inherit the undesired traits. So, there is no guarantee that the resultant hybrid progeny will definitely carry all desired characters. They may also carry undesired characters. For this reason, traditional hybridization is not considered as the most superior technique. But when we go for recombinant DNA technology of genetic engineering, in genetic engineering, we can pick the desired gene or a set of desired genes, and we can introduce only those genes specifically into the target organism. It is like hitting someone at a point blank range. So, if you shoot someone at a point blank range, it is going to directly hit his head without any chance of missing. And genetic engineering is such a powerful technique where you can pinpointedly pick the genes of your choice and you can place them, introduce them into the target organism. And in this case, there is no chance of introduction of undesirable genes at all. And that is the reason today genetic engineering has become one of the most powerful techniques in the fields of biology. Right? And dear children, that's all for the day. I think the conceptual clarity has come. Now you know what is the definition of biotechnology, what are the two core areas of biotechnology, and how is the recombinant DNA technology much more superior when compared to that of the sexual reproduction and traditional hybridization procedures. That's all for the day. In the next class, we will be discussing about the fate of an alien piece of DNA and the very first experiment that laid the foundation for the development of the field that is biotechnology. Thank you for the day. Goodbye.